Okay, excellent. So like Matt said, I'm going to be talking about it's fairly old work now, but since I'm a very lazy publisher, I haven't published most of this yet. I'm going to talk a little bit about perennial Lupinus in California and why there are either so many species or just one giant, very diverse species. A little bit of background about the genus overall is that it's a very diverse genus. It's a Fabaceae. The genus includes annuals and perennials and shrubs. It has a major diversity hotspot here in California. The genus Lupinus, conveniently though, is a natural taxonomic group. It's the only group in the Lupininae. Nearly all the lupins have these digitally palmate leaves, which makes them really easy to spot in the field. And lupins also have a common and unique pollination mechanism, which can be thought of as like a piston mechanism. A large insect like a bumblebee will sit on the wings and keel of the flower and push down on it, and pollen will push up. The pistil will push the pollen through the tip of the keel onto the bee, and that's how the pollen gets transferred. But if the flowers start becoming too small and things can't land on them anymore, or they just don't get pollinated, most lupins are facultatively selfing, and some of the smaller flowered ones are obligately selfing because the stigma will grow directly up through the pollen at the tip of that keel. So the perennial lupins in California were my specific group of interest overall. There's somewhere between 48 and 100 taxa recognized in this group, depending on who you ask. They're very morphologically diverse. There's a lot of species and varieties described in this group. Their habits range from cespitopes, little cushion plants, and matting decumbent plants to large woody shrubs. Some are well over the height of a grown man, 6 feet, 8 feet, 10 feet tall. They occupy a diverse range of habitats. You can find them in xeric environments, mesic environments, low elevation to high elevation, coast to the inland. But previous work in this group has not really resolved any kind of monophyly for any of the species we currently recognize in this group. I think a bunch of us around the same time when we all started our research in the mid-2000s had hoped that we did some molecular phylogenetics on this group that we would retrieve some kind of signal of monophyly for something in this group, and we really didn't, which was very frustrating. We're looking at a slightly different approach to this. We went back to looking at Charles Pepper Smith and previous workers' groupings of the perennials into tribes, which don't really have a current equivalent in it. They don't really have a good taxonomic name for that particular level, so I just call them tribes in quotes. And I'll just describe a couple of them that I was particularly interested in my research. The tribe Daisie I. This includes most of the woody bush lupins in California. This would include the silverbush lupin in the sands of the coast of California, Lupinus chamasonis, all the members of the Lupinus albifrons complex, which would be albifrons, Lupinus excubitus, Lupinus longifolius, Lupinus peinii. And these all share the common character of having these dense oppressed hairs on their leaves, which give them a silvery sheen, so they're sericeous leaves, which is, hence Daisie I. All flowers for this group have a completely reflex banner petal, and their wing petals fully enclose the keel petals. And they generally have woody above ground stems. The tribe Albicollis is another very widespread group. This includes Lupinus formosus and Lupinus albicollis. These flowers tend to have a more sharply pointed falcate keel that is not fully enclosed by narrow wing petals, which makes it easier to, that makes a good morphological character. And a lot of these plants also tend to re-sprout each year from underground structures, like codexes or rhizomes. Because they do sprout from underground structures, they tend to flower a little bit later in the year, so while they often share similar habitats to members of the Daisie I, they don't flower at the same time. The arboreae are mostly coastal species. The main one in this group is the yellow bush lupin, Lupinus arboreus. Its relatives like Lupinus variecolor, Lupinus rivularis, Lupinus littoralis, all really hybridize with arboreus in nature, and these species have generally been allied with each other by most authorities. I'm also going to mention an annual clade, which has usually been found to be sister to the perennials. This is the tribe including Lupinus nanus and Lupinus bicolor, which are very common annuals here in California. There are many species in this clade that have been argued about delimitations of for many years, including Lupinus pactylobus, Lupinus affinis. This group has a lot of integrating forms. It makes it really hard to tell where the species lines are in this group. And I just point this out because they make a good outgroup for my perennial taxa most of the time. So I did some nuclear phylogenetics for my dissertation in this group. Back in the day, in 2008, 9, 10, we didn't have this new next generation sequencing thing yet, so we had to be very careful about what we were going to sequence. One of the best nuclear genes to sequence in lupins turned out to be the cycloidea paralog that had been described by Helen Satirin in 2005 as having pretty high levels of variation, so a lot of us tried that marker. The cycloidea genes control bilateral floral symmetry, just as a nice tidbit. And conveniently, there are 2 copies of it in lupins, which means that we have 2 independent unlinked loci that we can use for these kinds of analyses. So for my dissertation work, I had sequenced 50 samples of 27 perennial taxa and some outgroups for these 2 nuclear genes. One of these, Legsite 1b, did not give a very highly resolved phylogeny at all. Pretty much all you can say is that perennials are monophyletic. They are sister to the Nanus group, but there's pretty much no well-supported branches and very little resolution in this group. But the Legsite 1a phylogeny had a lot more structure. The perennials, again, were monophyletic. You can see the sister group Nanus again, sister to the perennials. Well, kind of sister. They were a little bit of a paraphyletic grade here. But you can start seeing some of those tribes again that I've talked about appearing as either monophyletic groups or paraphyletic grades near them. While this phylogeny is, you can sort of see the tribes emerging, a more informative way of looking at it, or at least a more easy way to visualize this, is using a haplotype network approach. So this is the same molecular data that I just showed you in the phylogeny, except using it as visualized as a graph instead. So so each single line between 2 dots represents a single inferred nucleotide change. The colored dots represent actual specimens I sampled, and their colors vary by species. And if there's larger dots, it means that I had more than one sample with that exact same haplotype. Here you can see that the tribes are clearly separate from each other. The outgroups that represent other tribes have unique haplotypes that are not part of any of these tribes that I'm talking about, which is very nice. But if you want to start to resolve species within one of these tribes, probably the best thing to do would be to look at, say, one of these tribes, look within it, and maybe use population genetic methods within this larger group Sericii, measure things oversample things within and between species in this group and throughout their geographic range and see if any patterns emerge there. So I focused on the Lupinus albifrons complex. This is a really interesting group to me, mostly because in the literature you can see these massive fights going on between various authorities over time. For example, like Philip Munns and then Teresa in the Jepson manual had recognized multiple species in this complex. Lupinus albifrons, mostly in Northern California-ish, Lupinus excubitus, a Southern California-ish, and then Lupinus longifolius on the coast. But Dwayne Isley, when he did his treatment in 1998, he lumped everything into Lupinus albifrons and said he couldn't see any differences, really. I resurrected Lupinus payneii in a paper I wrote a few years back, which seems to have been synonymized into Lupinus excubitus in past works, but is unique enough, both morphologically, locationally, and genetically to be, I think, recognized as a species again. But yeah, all the members of the Lupinus albifrons complex share these similar floral and leaf characters, but pretty much any character you look at is highly variable within each of these taxa. And my favorite quote about this is from R.F. Hoover, who said that varieties can only be distinguished by emphasizing a single feature and ignoring the general appearance of the plant, which is not a very helpful taxonomic feature in this group. And so just as an example of one of these highly variable features, they're just the size of the plants themselves. They can range from decumbent cespitose, little guys that are just barely less than half a meter tall, to This one that's over, like this one, Lupinus panii, that's over 2 meters tall. You can see, you know, a 6-foot tall man standing next to one of them. And these apparently are smaller than the original, in the same population. This wasn't from the same population that Theodore Cannon originally described back at the turn of the 20th century. And when he described them, they were 10 feet tall with massive trunks, and those guys are gone. But they're still pretty impressive, even though they're not as impressive as then. So it's hard to believe that these are all in the same species, you know? That's a lot of height variation. So what I did was I just sampled populations of all of these taxa from throughout the state. For each, I considered it to be a population. If I found, I would collect one voucher specimen for that population, and then 6 to 10 individuals plants from around 50 meters of that voucher plant. The populations tended to be fairly isolated from one another, so there wasn't a lot of integrating between populations. They're very clumpy. So there's 19 populations that I sampled from throughout California. They centered around the transverse ranges. There's a lot of interesting variation there, but also, you know, I was based at Rancho Santa Ana Botanic Garden, which is in Claremont, so transverse ranges. I sampled again 6 to 10 individuals per population, and I sequenced 2 unlinked regions: a short chloroplast region for that natural lineal genealogy. You know, we often do chloroplast phylogenetics to do this kind of work, and a short segment of that like 1A variable gene for nuclear alleles and some diversity analyses. I could also add in other albifron samples that I had collected from the overall phylogenetic experiments. They weren't from, I didn't do population level sampling of these, but you know, they're definitely in the albifrons complex, and I had the material so I could add these into the analysis as well. I also added 2 outgroups from the Daisie I, Lupinus chamasonis and Lupinus lutefigianus. Those are those gray stars. They're both in San Luis Obispo County. So here's what I found. If you just look at the chloroplast pattern, I find it more useful, again, to use the haplotype network approach than the phylogenetic tree approach. The tree tends to have a little bit less resolution, and you can't tease apart these relationships quite so well there. So the chloroplast tends to evolve much more slowly, so this should capture, the chloroplast phylogeny should capture more ancient changes, things that have been kind of just been preserved over time. And when I did this, I found that there were 2 haplotypes that were shared by by a lot of populations. 7 populations shared one of those 2 haplotypes in the big circles in red. And then if you consider haplotypes are just one nucleotide change away from them, you can get a whole lot of the populations, and also a lot of those other individual samples that I did. So you can see that the red circles on the map throughout the range of California, they all share these same few very closely related haplotypes. And again, there's very diverse morphologies captured in this very small group of chloroplast haplotypes, from the little guys to the really tall ones. But there was also a lot of populations that had distantly related haplotypes. Interestingly, these very diverse chloroplast haplotypes tended to be centrally located in the transverse ranges in the eastern Sierra Nevada. So I'll go over a few of those in more detail here next. That Lupinus panii that I was talking about that had this very distinct chloroplast haplotype, and in my previous chloroplast phylogenetic work, and also, you know, seems to be distinctive in appearance. Came out separately in this analysis as well. These populations are centered in the Santa Susana Mountains and in the Simi Hills. And you know, they're pretty closely related to those common haplotypes, but they're not the same. But there's this very, very distant haplotype, which was identical in 3 populations of Lupinus excubitus. But interestingly, they're completely geographically disjunct. There's one from the San Gabriel Mountains, and then 2 in the eastern Sierra Nevada, and they shared exactly the same haplotype. There's also several intermediate haplotypes that were from somewhere in the middle. These are things like Figueroa Mountain, Fraser Peak, and I forget what else, Leaver Mountain. So I don't know what to make of these guys. They might be different enough to represent other species, depending on how you want to look at it. But that way lies madness, so I tend to not, I've just kind of not dealt with it yet. so you would think with those kinds of, with that kind of chloroplast information, with each population just generally having one individual, like one haplotype per population, that there's not going to be that much diversity in this group. But for the nuclear locus that I sequenced, I did find that there were actually a lot of diversity at nuclear loci. On average, 77% of the alleles that I sequenced were different from the others at that population. So there is a lot of genetic diversity per population in each of these groups. And so I was interested in comparing gene flow between these groups to see if lupins actually, if they're actually just isolated, genetically isolated from each other, and that's what's happening, or question mark. So I compared pairs of nearby populations to each other, and I found that using like isolation by migration methods, that gene flow is significant. The arrows you can see are weighted by the amount of gene flow that was detected between those two populations. And you can see that in pairs that were of the same taxon, according to that chloroplast phylogeny, there was detectable gene flow. But there was nearly no gene flow in between pairs of populations that were from different taxa. And this is nuclear data, so the chloroplast haplotypes might reflect real genetic isolation, and maybe speciation is occurring in these kinds of groups. So basically, how I would describe what's going on in my understanding currently of what's happening in Lupinus albifrons and the albifrons complex is that there must be some populations, most of the populations of Lupinus albifrons, that recently and quickly reached that widespread distribution throughout the California floristic province from some ancestral distribution in Southern California. In Southern California, though, they must have been there longer. There's populations that that's their ancestral origin of this group, but they're still genetically isolated from each other as well as from that widespread taxon, so there's a lot more diversity there. The populations that are less closely related in chloroplast haplotype have been genetically isolated for a longer time. Maybe those populations are undergoing selection for similar characters per habitat. So like in each little pocket, each little clumpy area, given whatever genes it's got, they might be undergoing selection for similar characters, like convergent characters across these little pockets across the transverse ranges, like maybe intense sunlight or particularly dry soil. But since the populations are isolated and are not sharing genes, these changes are going to be independently derived. So we're going to see a lot of similar evolution for similar traits happening over and over again without any reference to the other populations. So the best way to look at it is that the Lupinus albifrons complex is a species flock. Each isolated population or group of populations is pretty much just adapting to its own local habitat and becoming its own separate species without reference to any other populations. So therefore, if you find populations that look similar, they might not be that genetically similar, which makes taxonomy really hard and means that I haven't bothered to figure out how I would like to describe that. And that's pretty much it. This talk was based on my dissertation research at Rancho, so thanks to my co-author Elizabeth Fryer, and these are, and I think we have time for a couple questions.